Have you ever wondered how scientists can literally go in and rewrite the code of life? Well, today we're cracking open the Molecular Biologist Toolkit to see exactly how they read, copy, and edit DNA. So if you've got a biology exam coming up, think of this as your ultimate review session. Let's jump right in.
I mean, it sounds like something straight out of a sci-fi movie, right? The power to edit an organism's genetic blueprint. But this isn't fiction. This is real science. A set of powerful techniques that have totally revolutionized things like medicine and agriculture. The big question is, how in the world do they actually pull it off?
Well, the whole field is called genetic engineering, and it's pretty much what it sounds like. It's the process of deliberately changing an organism's DNA to get a specific desired result. And the applications, they are massive. We're talking about everything from fixing genetic disorders to making our food supply better and even manufacturing life-saving medicines like insulin.
So, to really get how this all works, we're going to follow the exact same workflow a scientist would use in the lab. First, you've got to isolate your raw materials and make more of them. Then, you analyze the code you've got, and finally, you use all that information to build something completely new.
Okay, so step one. Every single genetic engineering experiment, no matter how complicated it gets, starts with these two basic tasks. First, you have to get your hands on the DNA you want to study. And second, you have to make enough copies of it so you can actually work with it.
So right away, you hit your first hurdle: access. See, DNA isn't just floating around for the taking. It's locked away inside a cell, protected by a cell membrane and, in many organisms, a nuclear membrane too. You can't do anything with it if you can't even get to it. And the solution to that problem is our first tool: DNA extraction. This is a chemical process that basically blows open those protective membranes. The scientific term is lyses them. And then it separates the DNA from all the other stuff, like the histone proteins that keep it all wound up. And what you're left with is pure, workable DNA. Okay, so you've got the DNA out, but now you have a new problem. You probably don't have very much of it. A lot of the time, especially in fields like forensics, the sample you start with is incredibly tiny, way too small to run all the tests you need. You gotta make more, like a lot more. This is where one of the most revolutionary tools in all of biology comes into play: PCR, which stands for polymerase chain reaction. The easiest way to think about it is like a molecular photocopier, but for DNA. It can take one tiny little piece of DNA and amplify it, making billions of identical copies in just a couple of hours. It's a total game changer.
So how does this photocopier actually work? Well, it's all about a three-step cycle that just repeats over and over. First is denaturing. You crank the heat up to about 95 degrees Celsius, and that heat splits the DNA double helix into two separate strands. Second, annealing. You cool it down, which lets these little DNA snippets called primers find and bind to the specific spots you want to copy. And third, extension. You heat it back up a bit, and a special enzyme called Taq polymerase gets to work, building a brand new strand of DNA. And here's the genius part. Taq polymerase comes from bacteria that live in hot springs, so it can handle the crazy high heat of that first step. Just like that, one copy becomes two.
But the real magic of PCR is in the repetition. A machine called a thermal cycler runs this three-step cycle, 30 or 40 times. And because the number of copies doubles every single cycle, you get this incredible exponential growth. You start with one molecule, after 30 cycles, you've got over 1 billion copies. It's amazing. All right, so now we have millions, even billions of copies of our DNA. We're ready for step 2, analyzing it. How do scientists actually read the code and, just as importantly, compare different DNA samples to each other? Well, to analyze a really long strand of DNA, the first thing you need to do is chop it up into smaller, more manageable pieces. And for that, scientists use another tool borrowed from nature: restriction enzymes. Think of them as tiny, incredibly precise molecular scissors. They actually come from bacteria, which use them as a defense mechanism to chop up the DNA of invading viruses. Each enzyme recognizes a very specific sequence of nucleotides and will only cut the DNA right there, which gives you a predictable set of fragments.
So after you let your molecular scissors do their work, you're left with this jumble, this mixture of DNA fragments of all different sizes. That by itself isn't super useful. You need a way to organize them, to see what you've got. So how do you sort them? The answer is a really cool technique called gel electrophoresis. I want you to think of this as a molecular racetrack. It's a clever way to separate all those DNA fragments based on just one thing: their size. It uses a slab of gel and an electric current to get the fragments racing. Here's how the race works.
First, your DNA samples get loaded into little pockets or wells at one end of a slab of gel. Then, you turn on the power. Now, remember, DNA has a negative charge, thanks to all those phosphate groups in its backbone. So when you apply an electric current, all the fragments get pulled toward the positive end. But the gel itself is like a tangled net, a molecular obstacle course. The little, smaller fragments can zip through it pretty easily and travel far. The big, bulky fragments, they get tangled up and move much more slowly.
And what you get at the end is this unique pattern of bands, where each band is a group of DNA fragments of the same size. This pattern is unique to each individual's DNA, creating what we call a DNA fingerprint. This is huge in forensics. Investigators can compare the DNA fingerprint from evidence at a crime scene to suspects. And you can see it right here. The pattern from suspect 2 is a perfect match to the evidence from the crime scene.
Okay, so let's recap. We can get DNA out of a cell, we can copy it, we can cut it up, and we can sort the pieces. Now for the final and honestly the most exciting part. Using all these techniques to combine DNA from different sources to build something brand new. This is where it really starts to feel like the future.
What you create in this process is called recombined DNA. It's basically a hybrid molecule, a biological mashup. It's made by joining DNA from different organisms together, like say taking a human gene and sticking it into the DNA of a bacterium. You're creating a DNA molecule that has never existed in nature before. The whole process is actually pretty elegant. First, scientists use one of those restriction enzymes, our molecular scissors, to snip out a specific gene they want from human DNA. And here's the key. They use the exact same enzyme to cut open a small circular piece of bacterial DNA called a plasmid. The human gene then fits right into that opening in the plasmid. A new enzyme called DNA ligase acts as a molecular glue to seal it all up. Then that new recombinant plasmid is put back into a bacterium.
This is the famous process used to make human insulin for diabetics. Once that recombinant plasmid with the human insulin gene is safely inside a bacterium, that bacterium does what bacteria do best. It divides and divides and divides. And every new bacterium that's born has a copy of that plasmid, creating this huge army of tiny, living factories all churning out human insulin.
And any organism that's been modified like this to carry genes from a different species has a special name. It's called a transgenic organism. You probably know them by the more common name, genetically modified organisms, or GMOs. That little insulin-producing bacterium, that's a classic example of a GMO. And the potential uses for this are just incredibly broad. We've talked about medicine, but in agriculture, this technology is used to create plants that are resistant to pests, which helps increase how much food we can grow. It could be used to engineer organisms that can survive in really tough environments, or even to improve our food in other ways, like giving it a longer shelf life.
These two examples really show you the amazing range of this technology. On one hand, you have something like a tomato that's been engineered so it doesn't rot as quickly. On the other, you have bacteria that are producing life-saving medicine. It's wild to think that the same basic toolkit, cutting, pasting, copying DNA, makes both of these things possible.
So, there you have it. A peek inside the scientist's toolkit for manipulating DNA. From extracting and copying to cutting, sorting, and pasting, these techniques give us a level of power over biology that was unthinkable not too long ago. And that, of course, brings us to the biggest question of all. Now that we have the toolkit to edit the very code of life, what should we build next?